Hello guys, welcome to my YouTube channel. So today we are going to discuss the steps in glycolysis. So basically all the steps in glycolysis can be divided into two major phases. Number one, which is the preparatory phase, and later on we will see the payoff phase. Now the steps number 1 to 5 comes under preparatory phase, and steps number 6 to 10 comes under the payoff phase. Now why steps 1 to 5 come under preparatory phase? As you will see later on, here glucose is prepared for glycolysis by investing 2 molecules of ATP. That means what? You are activating glucose for subsequent glycolysis by investing 2 molecules of ATP. That's why in a way you are preparing glucose. So this steps from 1 to 5 is called as the preparatory phase. So the first step in preparatory phase is the phosphorylation of glucose. Here what happens? You take glucose and the ATP is added, and from ATP the gamma phosphoryl group of ATP is added to the 6th carbon at this position, and here the 6th carbon of glucose is phosphorylated to form the glucose 6-phosphate. Next step is a conversion of glucose 6-phosphate to fructose 6-phosphate. So this is basically a reversible isomerization step catalyzed by the enzyme phosphohexose isomerase, in which a glucose 6-phosphate gets converted into the isomer fructose 6-phosphate. Next step is the phosphorylation of fructose 6-phosphate to fructose 1,6-bisphosphate. So here again the ATP is invested. So this is the second step where ATP is invested. The first was in step number 1. So fructose 6-phosphate reacts with ATP, where the gamma phosphoryl group of ATP is transferred to carbon number 1 now in presence of the enzyme phosphofructokinase 1 PFK1, and fructose 6-phosphate as a result is converted into fructose 1,6-bisphosphate. Now if you notice, fructose 1,6-bisphosphate has two phosphoryl groups, one at carbon number 1 and the other at carbon number 6. Alright. So next step of glycolysis is the cleavage of fructose 1,6-bisphosphate into dihydroxyacetone phosphate and 3-phosphoglyceraldehyde. So here in presence of the enzyme aldolase, fructose 1,6-bisphosphate undergoes reversible aldol condensation to give the 3-carbon compound dihydroxyacetone phosphate and another 3-carbon compound glyceraldehyde 3-phosphate. Now one thing you note here, the carbon numbers 1, 2 and 3 of fructose 1,6-bisphosphate have been retained in dihydroxyacetone phosphate, and carbon number 4, 5 and 6 have been retained in glyceraldehyde 3-phosphate. The next step is the interconversion of triose phosphate. So why this step is necessary? Because only one of the two triose phosphates that have been formed earlier can be used for subsequent steps of glycolysis, and the triose phosphate that can be used only in the subsequent step of glycolysis is glyceraldehyde 3-phosphate. So now all the dihydroxyacetone phosphate that had been generated in the previous step needs to be converted into glyceraldehyde 3-phosphate in order to continue the cycle of glycolysis. So here what happens, dihydroxyacetone phosphate in presence of the enzyme triose phosphate isomerase gets converted into glyceraldehyde 3-phosphate. So now previously we could distinguish dihydroxyacetone phosphate by the presence of carbon number 1, 2 and 3, and glyceraldehyde 3-phosphate has carbon number 4, 5 and 6. But now due to this isomerization step, these two become indistinguishable. Why? Because now you have only glyceraldehyde 3-phosphate in the media. So from the step number 6 to 10 is basically the payoff phase. Why it is called as payoff phase? Because the ATP or the energy that you had invested for glycolysis will now start to pay off. That means here the energy that has been invested for glycolysis, you will get more amount of energy in the subsequent steps of glycolysis back. Alright, so oxidation of glyceraldehyde 3-phosphate to 1,3-bisphosphoglycerate. So here what happens, glyceraldehyde 3-phosphate reacts with inorganic phosphate in presence of NAD plus, which is converted into NADH plus H plus in presence of the enzyme glyceraldehyde 3-phosphate dehydrogenase, and it gets converted into 1,3-bisphosphoglycerate. Why bisphospho? Bis means 2 and phospho means 2 phosphates. Here, as you can see, this is one phosphate group and here there is another phosphate group. So all total you have 2 phosphate groups at carbon that carbon numbers 1 and 3. That's why this is called as 1,3-bisphosphoglycerate. So next step is the phosphoryl transfer from 1,3-bisphosphoglycerate to ADB. So what happens? 1,3-bisphosphoglycerate reacts with ADB, in which this phosphate group is transferred to this ADB. Alright. So this phosphate group is transferred from 1,3-bisphosphoglycerate to ADB. So what will happen? 3-phosphoglycerate will form, and because this phosphate has been transferred to this ADB molecule, ADB will now be converted into ATP in presence of the enzyme phosphoglycerate kinase. So here what happens, from a substrate 1,3-bisphosphoglycerate, which is a very high energy compound, alright, and this is actually an acyl phosphate compound. It is very high energy compound. It is an acyl phosphate compound. So from this type of high energy compound, you have ATP formation. So you have ATP formation from a substrate. That is why this step is called as substrate level phosphorylation. So remember, step number 7 is the step where substrate level phosphorylation happens. Substrate level phosphorylation, SLP. So next step is the conversion of 3-phosphoglycerate to 2-phosphoglycerate. So in this step, what happens? This is basically a sort of isomerization. This phosphate group, which is present at carbon number 3 position, this phosphate group is now moves to carbon number 2 position, and this OH group goes to carbon number 3. So here that is basically interchange of the functional groups, but the basic structure or the structural formula remains the same. So 3-phosphoglycerate is converted into 2-phosphoglycerate in presence of the enzyme phosphoglycerate mutase. So the second last step is the dehydration of 2-phosphoglycerate to Phosphoenolpyruvate. Here what happens? This OH group at carbon number 3 and this H group at carbon number 2, these two go out with the as water molecule, and as a result here the double bond forms in between them. This is called as Phosphoenolpyruvate. Here as you can see, En means double bond has been formed. So Phosphoenolpyruvate. And the last step is the phosphoryl transfer from Phosphoenolpyruvate to ATP. So this is another step, second step where ATP has been formed. So first step where ATP has been formed was step number 7, which was substrate level phosphorylation. So this is the second step where again ATP is being formed. So what happens? Phosphoenolpyruvate reacts with ADB, and this phosphate group is transferred to ADB. Alright, so what will happen? ADB will get converted into ATP and pyruvate will be formed. So in presence of the enzyme pyruvate kinase. Alright, so final product that we get from glycolysis is pyruvate, which will be utilized maybe in the Krebs cycle or in other types of metabolic reactions. So let us see the overall balance sheet of glycolysis. Here as you can see, you had invested 2 ATP molecules previously for charging glucose or for activating glucose. Alright, so you had invested ATP in glucose in step number 1 and in step number 3. Alright, so here 2 ATP and 2 NAD plus was also there, plus 4 ATP plus 2 PI gives you 2 pyruvate. So from 1 moles of glucose we get 2 moles of pyruvate. Alright, plus 2 ADP plus 2 NADH plus 2 H plus 4 ATP. So plus 2 H2O. So from 1 mole of pyruvate we had been getting 2 ATP molecules. So from 2 moles of pyruvate we are getting 4 ATP molecules. Alright, and what are the steps which we got the ATP molecules? This was step number 7. Okay, and step number 9. Sorry, step number 10. So These two are the steps in which we had got ATP molecules. Now as you can see, we had invested 2 ATP molecules and here we are getting back 4 ATP molecules. So the net gain in glycolysis is 4 minus 2 or 2 ATP molecules. So in glycolysis, the net gain of ATP is actually 2 molecules of ATP.